From the chapter Biomolecules, we are going to study some additional points about enzymes. Now, classification of enzymes. The enzymes, they are classified into 6 different types on the basis of the reactions they catalyze. The first type of enzymes are oxidoreductases. These are enzymes catalyzing oxidation and reduction reactions by the transfer of hydrogen and or oxygen. For example, alcohol dehydrogenase. Alcohol dehydrogenase is an oxidoreductase enzyme. Here, alcohol plus NAD in presence of alcohol dehydrogenase forms an aldehyde and NADH2. If here the alcohol is ethyl alcohol, so here acetaldehyde will form and NADH2 is given out. Then next are transferases. These enzymes catalyze the transfer of certain groups between two molecules. For example, glucokinase. Glucokinase enzyme transfers the phosphate group from ATP to glucose. So in this reaction, glucose plus ATP, adenosine triphosphate, in presence of enzyme glucose kinase forms glucose 6-phosphate. The 6th carbon of glucose gets the phosphate group from ATP and it forms glucose 6-phosphate, and ATP is converted into ADP, adenosine diphosphate. This is the first reaction of glycolysis. Here glucokinase is a transferase enzyme which transfers the phosphate group to glucose. Then next type of enzymes are hydrolases. These enzymes catalyze the hydrolytic reactions. Hydrolytic reactions are the reactions in which the water molecule it splits and the substrate also it splits up to form its constituents. So this class includes amylases, proteases, lipases, etc. For example, sucrase. Sucrase enzyme, it hydrolyzes sucrose into glucose and fructose. The sucrose is a disaccharide. It is broken down and it is converted into its monosaccharides glucose and fructose with the help of hydrolase enzyme sucrase. Similarly, amylases, which are present in saliva, they also break the food material and help in digestion of food. Proteases break the proteins and lipases break the lipids. Lyases, these enzymes are involved in the elimination reactions resulting in the removal of a group of atoms from substrate molecule to leave a double bond. It includes aldolases, decarboxylases, dehydratases, for example, fumarate hydratase. Then histidine, in presence of histidine decarboxylase, forms histamine and CO2 is given out. Here, from the histidine molecule, CO2 is removed in presence of enzyme decarboxylase and histidine is converted into histamine. Then isomerases, these enzymes catalyze the structural rearrangements within a molecule. Their nomenclature is based on the type of isomerism. Thus, these enzymes are identified as racemases, epimerases, isomerases, mutases. Example, xylose isomerase. Glucose 6-phosphate. It is converted into fructose 6-phosphate in presence of enzyme isomerase. This is one more reaction of glycolysis where glucose 6-phosphate is converted into fructose 6-phosphate in presence of enzyme isomerase. The next type of enzymes are ligases or synthetases. These enzymes catalyze the covalent linkage of molecules utilizing the energy obtained from hydrolysis of an energy-rich compound like ATP, GTP, for example, glutathione synthetase and pyruvate carboxylase. These are the examples of ligases. Now, pyruvate carboxylase, it catalyzes the reaction in which pyruvate plus carbon dioxide plus ATP, it forms oxaloacetate and ADP is released and the phosphate group is also released. Here pyruvate is a 3-carbon compound. It combines with carbon dioxide to form the 4-carbon compound oxaloacetate. This is an important initial reaction in many cycles of photosynthesis. Here, pyruvate carboxylase functions as a ligase or a synthetase which joins the pyruvate and carbon dioxide to form oxaloacetate. Mechanism of enzyme action. The basic mechanism by which enzymes catalyze chemical reactions begin with the binding of the substrate to the active site of the enzyme. The substrate binds to the active site of the enzyme. The active site is a specific region where the substrate it binds to the enzyme and it forms the enzyme substrate complex. The binding of substrate to the enzyme causes change in the distribution of electrons in the chemical bonds of the substrate and ultimately causes the reactions that lead to the formation of products. The products are released from the enzyme surface to regenerate the enzyme for another reaction cycle. There are 2 models to explain the mechanism of forming enzyme substrate complex. The first model is the lock and key model. This model, it was proposed by Emil Fischer in the year 1894. The enzyme, specific action of enzyme with a single substrate can be explained using the lock and key model. In this model, the lock is the enzyme and the key is the substrate. Only the correctly sized key substrate fits into the keyhole active site of the lock enzyme. Here enzyme is the lock, active site is the keyhole and substrate is the key. The particular key only fits into the particular keyhole and it forms the enzyme substrate complex. After the products are released, enzyme is set free and it attaches to the new substrate molecule. The next model is the induced fit model proposed by Koshland in 1954, which states that approach of a substrate induces a conformational change in the enzyme. It is more accepted model to understand the mode of action of enzyme. Unlike the lock and key model, the induced fit model shows that enzymes are rather flexible structures in which the active site continuously reshapes it by the interactions with the substrate until it is completely bound to the enzyme. Here this enzyme molecule, it is not fixed but it is flexible, and as the substrate it approaches the enzyme, there are certain changes in the enzyme active site until the substrate it fits the enzyme and forms the enzyme substrate complex. Afterwards, the product is formed and the enzyme is released. Now we will study the factors which affect the enzyme activity. Following are the factors. The first factor is the concentration of substrate. The concentration of substrate, when plotted with the velocity of enzyme activity, we get a hyperbola curve. A rectangular hyperbola is obtained, and there are three distinct phases A, B, C of the reaction in the graph. Here V is the measured velocity of enzyme activity, Vmax is maximum velocity, S is the substrate concentration, and Km is the Michaelis-Menten constant. Km, or the Michaelis-Menten constant, it is defined as the substrate concentration expressed in moles per liter. This is the substrate concentration which is required to produce the half maximum velocity in the enzyme catalyzed reaction. Half maximum velocity of enzyme activity is reached at this substrate concentration. It indicates that half of the enzyme molecules are bound with the substrate molecules when the substrate concentration equals the Km value. The Km value is constant and is characteristic feature for a given enzyme. It represents a enzyme substrate complex. A low Km value indicates a strong affinity between the enzyme and the substrate, whereas a high Km value reflects a weak affinity between them. For majority of enzymes, the Km values are in the range of 10 to 5 to 10 to 2 moles. 10 raised to minus 5 to 10 raised to minus 2 moles. Next factor is the enzyme concentration. The rate of an enzyme reaction is directly proportional to the concentration of the substrate. The rate of the reaction is also directly proportional to the square root of the enzyme concentration. It means that the rate of reaction increases by the increasing concentration of enzyme. Now here you can see that in the graph, if there is no enzyme, the product concentration is very less. When with the increase in enzyme, the product concentration it increases, and with the doubling of enzyme, the product is more formed. This indicates that when the enzyme is increased, the rate of product concentration or rate of enzyme it increases. Temperature. The enzyme activity is best at the optimum temperature, which is 40 degrees Celsius. Below the optimum temperature, the rate of reaction slows down, and above the optimum temperature also, the activity slows down. Effect of pH. The rate of reaction is maximum at the optimum pH. The pH is different for every enzyme. Every enzyme has a different optimum pH. The enzyme cannot perform its function beyond the range of this pH value. Other factors that affect the enzyme activity are the increase or decrease in the presence of some substances like coenzymes, activators, and inhibitors. Coenzymes are the organic compounds which are required for the enzyme activity. Activators and inhibitors are also important. There are some apoenzymes also which are responsible for the enzyme activity. Thank you.